Now that we've finished with the first two organic molecules, we are going to get into proteins. Proteins contain carbon, hydrogen, oxygen, just like carbohydrates and lipids. However, they add another element, nitrogen, which is abbreviated N. The subunit of a protein is an amino acid, and amino acids are also called peptides. Proteins function as structural molecules and help control chemical reactions in cells. We are going to talk about that part later where it says help control chemical reactions in cells. That is what enzymes do, which is a category of proteins all by itself. So there's a couple of vocabulary words that you need to familiarize yourself with. The first one is peptide. I just told you peptide is another name for an amino acid. A peptide bond is what we call the bond between two amino acids. After dehydration synthesis occurs, there's going to be a bond between the two amino acids that is called a peptide bond. A dipeptide is two amino acids bonded together, just like a disaccharide is two carbohydrates bonded together. And a polypeptide is many amino acids bonded together, just like a polysaccharide is many carbohydrates bonded together, specifically many monosaccharides bonded together. Before we get into talking about proteins in general, we need to talk about the structure of an amino acid. You guys learned the structure of the individual monosaccharides, the glucose, fructose, and galactose. And you also learned the structure of glycerol, and you learned the structure of fatty acids. Just like you did those, you need to learn the structure of an amino acid. There are only 20 different amino acids that exist in nature. If you think of amino acids like the letter of the alphabet, there are 26 letters in the alphabet, but look how many different words we can make. If you try to picture it like that, there's only 20 amino acids in nature, but depending on how those amino acids are combined, you can make thousands and thousands of different proteins. Each amino acid has a central carbon atom with the following four groups attached. So there is a central carbon, meaning the carbon is in the very middle of the molecule. As you know, carbon bonds to four other things, whether they're atoms or other molecules. So in the case of an amino acid, it bonds to an amino group, which is abbreviated NH2, and I'll show you the structure of that. A carboxyl group, which you guys already learned when we did lipids. A hydrogen side group, which is nothing more than just a hydrogen atom attached to the central carbon. And then there is this R group. The R group is any number of atoms or molecules, and it is what gives the amino acid its unique properties. In my class, I say the R stands for random. Different classes say that it stands for different things, but nonetheless, whatever you want to call it, it's an R group. Now let's look at what those actually look like. So, here we have the central carbon. Attached to the central carbon is an amino group, which we abbreviated NH2, a carboxyl group, which we abbreviated COOH, a hydrogen side group, which is just an H, it's a hydrogen atom, and the random group, which is the R. So each one of those attaches by a single bond to the central carbon. Here are some examples of amino acids. If you see, here's the central carbon. Here is the amino group, the carboxyl group, the hydrogen side group, and the R group. Here, it's simply a CH3. This is the R group on valine, leucine, isoleucine, phenylalanine, and this strange thing here is the R group on proline. So, as you can see on each one of these, the central carbon, the amino group, the carboxyl group, and the hydrogen are exactly the same. It's only the R group that changes. Now we're going to talk about the dehydration synthesis of amino acids to form proteins. Dehydration synthesis is also called condensation. Those words are used interchangeably. It is slightly different than the condensation of carbohydrates and lipids. The reaction happens between the hydroxyl group of the carboxyl group and one of the hydrogens of the amino group. I actually think it's a much easier process to do than it was in either the carbohydrates or the lipids. So hopefully you guys will find it easier as well. Because of where the reaction is happening, a peptide bond forms between the carbon in the carboxyl group of one amino acid and the nitrogen in the amino group of another. You're going to see when I show you in just a minute, there is no oxygen in the middle. You are simply taking out a hydroxyl group from the carboxyl group and a hydrogen from the amino group. It's not a hydroxyl hydrogen, it's just a hydrogen that's all by itself. So when you form water, there isn't an oxygen left behind like there was on the previous two organic molecules. So this is one amino acid. This is another amino acid. Here's another one and a fourth one. We're going to take the hydroxyl group from the carboxyl group of this amino acid and the hydrogen from the amino group of this amino acid. We're going to take that water out, and then we're simply going to draw a bond between the carbon and nitrogen, which would be directly from here to here. You do that same thing 2 more times, and you end up with this protein plus 3 waters that you took out. 1, 2, 3. I don't know about you guys, but I find that much easier than the dehydration synthesis of either carbohydrates or lipids. The last thing we are going to discuss is called the level of protein structure. There are 4 levels. They are named primary, secondary, tertiary, and quaternary. We are going to do them each individually. It's pretty simple stuff. The primary level simply tells you the sequence of amino acids. So if you look here, I know you guys don't know these names yet, but there's glycine, leucine, valine, lysine, glycine. Maybe that's, I don't know, I can't read that. Alanine, lysine, valine. The order that these amino acid goes in tells you the primary structure. It is the amino acid sequence. The secondary structure is the coiling and folding of the primary structure, and the secondary structure can be in either an alpha helix or a beta sheet. This one is an alpha helix. A helix is something that looks like a spring. A beta sheet, I'm going to show you on the next page, but it looks like one of those paper fans you used to make when you were in elementary school. So this is an alpha helix looking like a spring, and this is a beta sheet. If you can see, it's folded on itself like an accordion, kind of. The tertiary structure is the secondary structure further folded upon itself, it looks like that, just like a jumbled up mass. This here is the alpha helix right in the middle. And you can see the alpha helix is then folded further onto itself. And then the quaternary structure is 2 amino acid chains folded together. So we have 2 different ones on this paper. If you look at these, here's the primary structure. It is the sequence of amino acids. Here's the secondary structure, which is the primary structure coiled or folded. Alpha helix looks like a spring. The beta sheet looks like an accordion or like one of those folded paper fans. Tertiary structure is the secondary structure folded upon itself, and the quaternary structure are 2 or more amino acid chains folded together. This is just a different diagram of what the different structures look like. And this is what an actual protein would look like. So those are your notes on proteins. I hope you understood everything. There will be a warm-up tomorrow on proteins. Have a great rest of the night.